So the next important topic of principle of inheritance chapter is pedigree analysis. So from this they will ask you definitely one question. Okay, so they will give you pedigree and they will ask this pedigree is related to autosomal dominant disorder or X-linked or autosomal recessive or X-linked recessive disorder. Okay, so what is pedigree analysis? So pedigree, it is the representation or chart, okay, showing, it is representation or chart showing family history. Okay, so pedigree is the representation or a chart that shows family history, and pedigree, it is important to study, right, the genetic disorders. If there is genetic disorder in any family tree, okay, or in any generation, if there is genetic disorder, that can be cured or that can be diagnosed by pedigree analysis. Okay. So in pedigree analysis, circle represents female, square represents male. Okay, circle female, square male, and if this circle is colored, okay, so that female is affected. So this is affected female, and if this square is colored, so affected male. Okay, and if there is circle and square in this fashion, so then there is mating of male and female. Okay, so this represents mating, right? So next next is, if in pedigree, if this chart is given, means, so this is mating, and this mating results into production of 3 individuals. So this is generation 1 and this is generation 2. Okay, so to solve any pedigree chart, okay, so you have to remember only these things, these 3 or 4 things. Okay, so I am giving you one example of pedigree and I will show you how to analyze the pedigree, how to solve the pedigree. Okay, so if this kind of pedigree is given, okay, where in every generation, this is generation 1, this is generation 2, and this is generation 3, this is generation 3, and this is generation 4. Okay, so how can you detect if this pedigree is X-linked or autosomal? right? So first, how to detect whether pedigree is autosomal dominant or autosomal recessive? Okay, so if the disease is present in every generation, okay, if the disease is present in every generation, that is dominant case, autosomal dominant case. Okay, so your disease is present in generation 1, but disease is absent in generation 2. In generation 2, there is no individual which is diseased. It is present in generation 3 and it is also present in generation 4. So this kind of pedigree is autosomal recessive. Okay, this kind of pedigree which is lacking one generation. Okay, in one generation if there is no diseased individual, that is autosomal recessive type of pedigree. Okay, for example, if this individual is diseased, right? So here generation 1 they have individual which is diseased, generation 2, generation 3, generation 4. So here in all generation disease is present. Okay, so this kind of pedigree is autosomal dominant. Okay, right? If disease is present in all generation, autosomal dominant. If disease is skipping any one generation, then disease will be, the pedigree will be autosomal recessive. So example of autosomal dominant, you all know there is NCRT, myotonic dystrophy or muscular dystrophy. Example of autosomal recessive is sickle cell anemia. So we will discuss sickle cell anemia in Mendelian disorders. Okay, so next, after pedigree analysis, we will see type of disorders related to chromosome. Okay, so the last topic of this chapter, genetic disorders. Okay, so what are genetic disorders? So the disorders which is related to increase or decrease in, we can say, genes, DNA or chromosome. Okay, if the individual has more number of chromosome or more number of or less number of or reduced number of chromosome, it will lead to genetic disorder, right? So this genetic disorder is divided into two types. So first one is Mendelian disorder. And second is chromosomal disorder. Okay, Mendelian disorder and chromosomal disorder. So Mendelian disorder, what is Mendelian disorder? So this disorder, they are caused by mutation of single gene. Okay, if there is mutation of single gene, okay, so whenever there is mutation of single gene, that will lead to Mendelian disorder. And when there is increase or decrease in whole set of chromosome, that will lead to chromosomal disorder. So first of all, what is gene? What is gene? So gene is the functional segment of DNA that codes for protein. Okay, so if for example this is DNA, okay, this is DNA, and for example this is the functional segment which will produce RNA and then protein after translation. Okay, right? So for example, if this part will undergo transcription and produce functional mRNA, and after translation they will produce functional protein. Functional protein, remember, right? So that functional part or that functional segment of DNA is known as gene. So there are hundreds, millions of genes are present in human DNA, right? So genes In genes, nucleotides are present, A, T, C, G, right? These four nucleotides are present. So A, T, C. So T, A, T. So complementary will be present on another strand, A, G. Okay. So whenever there is mutation in any of these nucleotide, right? So this mutation can be point mutation or frame shift mutation, right? So whenever these nucleotides are removed or some another nucleotide is added in gene, it will lead to Mendelian disorders. Okay. So first Mendelian disorder, which is given in your NCERT, is hemophilia. Okay. Hemophilia is there, sickle cell anemia, thalassemia, color blindness, okay, color blindness, cystic fibrosis, phenylketonuria, okay. But in NCERT, you have to study hemophilia, sickle cell anemia, right? Color blindness, okay. So remember, out of these 1, 2, color blindness 3, okay. So thalassemia is also there in your syllabus. Hemophilia, okay, and color blindness. Hemophilia and color blindness. These two are X-linked disorders. Okay, hemophilia and color blindness. These two are X-linked or sex-linked recessive disorder. What is X-linked or sex-linked recessive? So there will be presence of extra X chromosome, okay, or there will be mutation on X chromosome, right? Linked. Okay. So first one is hemophilia. Hemophilia is X-linked or sex-linked recessive disorder. Okay, so what happens in hemophilia? So there is a protein we all know which involved in blood clotting. Okay, fibrinogen. Okay, so there will be less production of that fibrinogen protein that will inhibit the clotting. Right. So this condition is hemophilia. So in hemophilia, clotting factors are reduced, or clotting factors are decreased. So that will inhibit clotting in blood. So whenever there is cut, the blood will flow. Okay. So there will be no clotting. If there will be no clotting factor, so the blood will continuously flow. Okay. And the clotting factor which is present in blood, that is fibrinogen. Right. So there will be excessive loss of blood due to hemophilia, hemophilic condition. Okay. So what happens in hemophilia? So these are X-linked, means in female we all know 2 X chromosome is present. Okay. In male 1 X chromosome is present. Okay. So if the gene for hemophilia, if the gene for hemophilia or the gene which is causing hemophilia, if one gene is present on one X chromosome of female, then this will lead to carrier female. Carrier female. If there will be one hemophilic gene on one X chromosome of female, it will lead to carrier female. If one hemophilic gene is present on X chromosome of male, it will lead to hemophilic male. Hemophilic male, right? And if both X chromosome of female is having this disease or hemophilic gene, right, it will lead to hemophilic female. Okay, hemophilic female. So XH hemophilic female, XHY hemophilic male, XHX carrier female, XY normal male, XX normal female. Okay. So here remember hemophilia. In females, this hemophilia is very rare. Okay. In female, this hemophilia is very rare because it happens only when mother is infected. Okay. Or mother is carrier or father is hemophilic. In that condition only the female child will be hemophilic. So father should be affected. Okay. Father should be affected. If father should be affected, then only their daughter will be affected. Okay. So we'll see one example. We'll see one pedigree. Okay. So in generation 1 the father is affected and mother is carrier, right? So what will be their autosomal condition? So X hemophilic Y, X hemophilic X. Okay. So daughter will be hemophilic XH. Okay. XHX means one daughter will be carrier. Okay, carrier, right? XH and this X. XHY, one male will be affected. Okay. XHY and this male will be normal. Okay. If this female is carrier, okay, if this female is carrier and if this male is normal, so what will be their progenies? If there will be, so there will be hemophilic allele in the progeny, yes or no? Yes. So here female will be carrier XH. So in this generation one female will be carrier and male will be 50% male will be affected. XX. XHY. Okay, XHY. So 50% of male will be affected and 50% of male will be normal. Here also 50% female carrier and 50% female normal. Okay. So this is about hemophilia. So next is color blindness. So color blindness is also X-linked or sex-linked. Recessive disorder. Okay, so it is same as hemophilia. Their genetic pattern is same as hemophilia, but in color blindness, what happens? So the infected individual, they can't differentiate between red and green color. Okay, so in color blindness, the infected individual, they loses their ability to differentiate between red and green color. Okay, and it is similar to hemophilia, where the infected allele or infected gene is present on X chromosome. Okay. So carrier female, XX, hemophilic female, color blind female, XCXC, color blind male, normal male, normal female. Okay, so XC, carrier female, carrier for color blindness. There will be no symptoms of color blindness in carrier female. Color blind female, XC, so both gene will be present on both chromosomes. Both infected gene will be present on both X chromosomes. XCY, color blind male. And this color blindness, it occurs in 8% of male and 0.4% of female. Okay, 8% of male is affected by color blindness and 0.4% they affect to female. Okay, because if female is affected, that means male parent, okay, should be affected and female must be carrier. Okay, female parent must be carrier and male parent must be affected. So that's why both affected X chromosome will pair up in fertilization. Sickle cell anemia is, they are controlled by two alleles, HbA and HbS. So these two alleles, they control sickle cell anemia. So whenever in a person HbA is present and HbA is present, that means the person is normal. Whenever these two alleles present, so that person is normal. So whenever HbA and HbS alleles are present, means that person is carrier for anemia. Okay, it can be male or female. Okay. So whenever HbS and HbS condition is present, this means the person is anemic. Okay, means he or she is suffering from ammonia. So here this alleles, it can be present in male or female. Okay, and sickle cell anemia, they are autosomal recessive type of disorder. Sickle cell anemia is autosomal recessive disorder due to change in nucleotides in autosomes result into anemic condition. Okay, so the next disorder is phenylketonuria. Phenylketonuria is quantitative disorder in which quantity of hemoglobin or RBC is reduced. Okay, phenylketonuria, it is also autosomal recessive disorder. It is also autosomal recessive disorder. So what happens in phenylketonuria? So there is an enzyme, okay, which converts phenylalanine into tyrosine, okay, and that enzyme is phenylhydroxylase, okay, phenylhydroxylase. Phenylalanine hydroxylase. So this enzyme, it converts phenylalanine to tyrosine. So tyrosine is an amino acid. Okay. So phenylalanine is converted into tyrosine by this enzyme phenylalanine hydroxylase. So in phenylketonuria, this enzyme production is decreased due to mutation. Due to mutation, this enzyme production is decreased so that the conversion of phenylalanine to tyrosine is reduced. So this will cause accumulation of phenylalanine in blood or tissues. Okay. So whenever there is absence of this enzyme, this phenylalanine, they will start accumulating in our body and they are excreted out by urine. So by urine analysis, we can check if there is presence of phenylalanine or not. Right. So this helps in diagnosis of phenylketonuria. Okay. So whenever there is large amount of phenylalanine is accumulated in our body, it start moving into our brain and cause mental disorders. Okay, and this phenylketonuria, their autosomal recessive disorder. Okay, this thing is in phenylketonuria. And one more thing, this phenylalanine, right? They start converting into pyruvic acid. Okay, so there is one enzyme present in human body, in liver, that converts phenylalanine into pyruvic acid, and this pyruvic acid, they also start accumulating. So they cause mental disorders whenever they move to brain. Okay. The next disorder, this is also a type of Mendelian disorder. Next is thalassemia. So thalassemia, there are of two types, alpha thalassemia and beta thalassemia. Alpha thalassemia and beta thalassemia. So what happens in alpha thalassemia? There is reduction of alpha globin of hemoglobin. Okay, and in beta thalassemia, beta globin is reduced in hemoglobin. So we all know hemoglobin, they are made up of, so hemoglobin which is present in RBC, they are made up of 2 alpha chain and 2 beta chain. Okay, so whenever alpha chain is affected, so there will be reduction in alpha globin. If beta chain is affected, there will be reduction in beta globin. So it will lead to alpha globin thalassemia or beta thalassemia. Right. So they are also autosomal recessive disorder. Thalassemia is autosomal recessive disorder. So it is similar to sickle cell anemia, but here the quantity of RBC is reduced. Okay, so they are quantitative disorder. So in both sickle cell anemia and thalassemia, production of RBC is affected. Okay, but in sickle cell anemia, the quality is reduced. So they are qualitative disorder. In thalassemia, the quantity is reduced. The quantity of RBC is reduced. So it is known as quantitative disorder. Okay, and this quantity is decreased by mutation. Okay, by mutation. So mutation in chromosome which produces alpha protein and beta protein. Okay, so if there will be mutation which is producing these two proteins, it will lead to thalassemia. Okay, this thing you remember in disorders, Mendelian disorders. Okay, so the next disorder after Mendelian is chromosomal disorder. So in chromosomal disorder, we will see there is increase or decrease in set of chromosome, and that chromosomal disorder will lead to 3 major kind of disorders, that is Turner, Down and Klinefelter syndrome. So chromosomal disorder, so whenever there is increase or decrease in chromosome, or a whole set of chromosome is increased or decreased, that is known as chromosomal disorder. So in chromosomal disorder, two things is there: aneuploidy okay. So increase or decrease of chromosome or set of chromosome. So when a chromosome, they occur in pairs. Okay, when whole set is increased or decreased, it will lead to chromosomal disorder. So two things is there in chromosomal disorder: aneuploidy and polyploidy. So what is aneuploidy? So aneuploidy, so this is whenever there is gain or loss of chromosome. Okay, whenever there is gain or loss of single chromosome. So whenever there is gain or loss of single chromosome during a cell division, okay, so whenever there is gain or loss of single chromosome during cell division, it gives rise to aneuploidy, right? Okay, so what happens in cell division? In anaphase, when chromatids are separated, when chromosomes are separated, okay, so when there is failure in chromatic separation during anaphase of cell division, okay, whenever there is failure, it will give rise to aneuploidy. Okay, it will give rise to aneuploidy. So aneuploidy, they are generally of 4 different types. Okay, so based on increase or decrease in chromosome during separation. The first is monosomy. Monosomy, when single chromosome is increased. Nullisomy, trisomy and tetrasomy. Okay, monosomy, nullisomy, trisomy and tetrasomy. So what happens in monosomy? So when one chromosome is decreased, 12 is deployed, we are deployed, and during cell division, when one chromosome is decreased during segregation process in cell division, it will lead to monosomy. Nullisomy, when two chromosomes are decreased, it will lead to nullisomy condition. Trisomy, when one chromosome is increased in any cell, okay, during cell division, during chromatic separation, it will lead to trisomy. Tetrasomy, when two chromosomes is increased during cell division in any cell, it will lead to tetrasomy. Tetra, 2, 2, 4, tri, 2 plus 1, 3, nulli, 2 minus 2, 0, monosomy, 2 minus 1, 1, 1, mono. Okay, so in this you can remember this. Right, so this is about aneuploidy. Okay, so next we will see what is polyploidy. So polyploidy, so during cell division, when there is failure in segregation, so this will result into increase or decrease of whole set of chromosome. So when whole set of chromosome is increased or decreased during cell division, this will lead to polyploidy. Okay, so not decrease. Okay, an increase of whole set of chromosome. Increase of whole set of chromosome leads to polyploidy condition, right? And this polyploidy condition is very rare in humans. This condition is only seen in plants. Okay, remember, polyploidy condition is rare in human. Okay, it is seen in plants, and this polyploidy condition will lead to 3 different type of disorders. Right? This polyploidy, they are also known as euploidy. Don't confuse. Okay, euploidy, also known as polyploidy. Okay, euploidy. So, chromosomal disorders, according to aneuploidy and polyploidy, there is 3 major disorders. First is Down syndrome. So Down syndrome is due to trisomy of chromosome number 21. Due to trisomy of chromosome number 21. So we all chromosome occurs in pair. Okay, chromosome occurs in pair. So in normal person, the chromosome number 21 will have 2 chromosomes. Okay, but in Down syndrome patient, that chromosome number 21, they have 3 chromosomes. So 3 chromosomes are present in Down syndrome patient in chromosome number 21. Okay, and this can be present, this syndrome, they generally affect children below 10 years of age. Okay, and their symptoms is important. So from this Down syndrome, either they will ask you whether it is trisomy of 21 or 24 or 26, okay, or they will ask you what are symptoms. So symptoms: mental retardation, short saturated head, okay, furrowed tongue, right? So these are the symptoms of Down syndrome. Okay, and remember their genetic constitution. So we all know a normal person 44 plus XY chromosome is present in male and 44 plus XX in female. Okay, but in Down syndrome patient, this number, this 44 will become 45. Okay, if it is in male, so 45 autosome, okay, and XY. In female, to 45 autosome plus XX. Okay, because there is trisomy increasing one autosome. Okay, so their genetic constitution is important, they might ask. Right, so this is about Down syndrome. Next syndrome is Klinefelter, which is generally seen in male. Klinefelter syndrome. So what happens in Klinefelter syndrome? So there is increase in one allosome. Okay, so we know allosome is X and Y chromosome. So there is increase in one allosome. So 44 autosome and allosome X, Y. This is normal constitution, but in Klinefelter there one more X chromosome will be present. Okay, and generally this Klinefelter syndrome, their patients are males. Okay, their patients are males. So generally this syndrome takes place in males. So their genetic constitution will be 44 plus XXY. Okay, 44 plus XXY. So one common symptom is there in Klinefelter, that is gynecomastia. That is enlargement of breast in male. Okay, enlargement of breast in male. Sterile male, whenever there is Klinefelter syndrome, male will be sterile. And mental retardation in patient. Okay. So this is about Klinefelter syndrome. So the last syndrome of chromosomal disorder is Turner syndrome. Okay, and generally Turner syndrome, their patient is only females. Okay, so what happens in Turner syndrome? So there is loss of one X chromosome, one allosome is lost in Turner syndrome patients. Okay, generally female they have 44 plus XX condition. Okay, normal female. So in this Turner syndrome they will have 44 plus X0 type of constitution. Okay, the total number of chromosome will be 45, 44 autosome and one allosome. Okay, 45. Klinefelter 44 plus XXY means 44, 45, 46, 47. Total 47 chromosome will be there in Klinefelter syndrome. Okay. So here symptoms will be sterile female. Okay, rudimentary ovaries means this ovary they will not produce eggs. Okay, so this thing are there in Turner syndrome. Mentally retarded dwarf. Okay, so mental retardation is there in all three syndromes, right? So this is the last topic of chapter. So if you have any doubt regarding this chapter, you please comment